Okay, good morning to everyone. First of all, I would like to thank Dean Thanapat and also the from Ransit University Faculty of Pharmacy and also organizing committee for Ransit University for inviting me to be a keynote speaker for this very special conference, the virtual conference. So today I'm going to talk about bioactive constituents from fermented noni, exudates, and antimicrobial activity of Hawaiian marine algae. Maybe a little bit ambitious, talk about two things at the same time. Hopefully we were able to finish up. So I would like to show this is the logo of our College of Pharmacy. Basically, this represents the volcano because that is in the the university is located in the active volcano, and usually in the College of Pharmacy in United States they use this snake as a symbol. So, give you a little bit background about if you have not been to Hawaiian Island. As you can see in this map here, this one, the most northern side are we call Kauai, and the one in the next one that you can see in this between these Pacific Oceans, the island is called Oahu. So most of you have heard about that. When you like to come to Hawaii, usually the airport that gonna allow you to do international transit is called Honolulu. So it's located in this island. The next island you can see is Molokai and Lanai. Those are smaller islands, and Lahainai and also Maui. So interestingly, the next one, which looks like a leaf, is we call it Big Island or we call it Hawaii. So this is where the active volcano is located. If you heard about the eruption happening that time, so it's the Kilauea volcano eruption here. So the Hilo town is here and the Kona is the other overseas in the west side. So the College of Pharmacy is built in this Pacific Ocean where we have this College of Pharmacy, University of Hawaii Hilo. That's what current is. So the next slide, I give you a little bit bigger picture. Think about when we talk about in Hawaii, if we want to study the natural product in Hawaii and the potential Earths, what are these about? So you can see in this diagram here, the upper part of some of the terrestrial plant and the lower part is some of the marine, the marine ocean plants. Okay, so let's start with the top panel first. The top one is the noni fruit. I'm originally from Malaysia. We also have noni fruit in Malaysia. I'm sure you also have noni in Thailand, so it might not be differences here. So the next plant you can see here is called Uhaloa. This plant we also study currently. It has a lot of alkaloid and other interesting natural products. The native Hawaiian usually use it for skin infection and for a lot of inflammation condition. In fact, the native Hawaiian healer depends on it now to produce as an anti-inflammatory agent combat the COVID-19. So the next plant, as you can see here, this plant is only unique in Hawaii. You'll not find in other places. It's called mamaki plant. This plant has full of antioxidant. They already make a tea. This tea is very effective for individual use it to reduce the blood pressure. So this is safe to drink and you can drink it as much as you can. So the next one is called tea leaf. They also has some spiritual talking about it, but there's definitely some important medicinal use of this plant. And also the next one is called kui nut. Interestingly, kui nuts has some antiviral activity. What happened is they use the nut and they have to take the fresh nut and then from the nut, from this kui fruit, they also a seed. They just need to get small amount of the sap. And this sap is very effective as a herpes and antiviral activity. Whether it has COVID-19 activity, nobody know yet. So again, we need to prepare this fresh sample and to test it. So the next one is called the ohia plant. It's red in color. It's kind of like common in the volcano area. The next one is popolo. So because as I showed you earlier, in ancient time for the Hawaiian, if they have to survive in this kind of environment, they have to depend on terrestrial plant and also the marine plant. So the Hawaiian, most of them, they eat fish and they eat poi. Now what is poi? Poi is called a taro. So because of that, they make good use of the environment. So they can harvest a lot of these algae. So some are called red algae, some are called green algae, some are called brown algae. In the Polynesian Hawaiian unique use these kind of algae, and they are edible one they call limu. So limu was the third component of the nutritional balanced diet consisting of fish and boil. If you come to Hawaii, you will see it's very common in the store. They actually prepare this fish, they call ahi. They eat the raw fish, they call it poki. They sprinkle with this red algae, and the name's called limu hoku. Okay, as you can see, they only use small amount. There are definitely some active ingredient in this red algae. Another algae that is edible is called the ogo. So, and as you can see in this diagram here. So towards the end, when we talk about the other project, we will see it's just the beginning of the project. So this next slide, I want to introduce you to some terminology called la'au lapa'au. So in Thailand, you have traditional medicine. In China, you also use traditional Chinese medicine. So in Hawaii, they also use traditional medicine, and they call la'au lapa'au. These are the pronunciation and also the way they uh, the way that they use that language this way. So Noni, the scientific name is called Morinda cingifolia. I think you already know that is you also have this plant in Thailand. In Hawaii, they use this plant in ancient for what? For diabetes, for asthma, for heart problem, for high blood pressure, for respiratory problem, inflammation and pain, a lot of variety. When they use the plant, they don't just use the fruit. They use the whole plant. They use the leaf for different purpose. They use the flower. They use even the seed. I think even the root. However, if they want to use the fruit, they only use the ripe fruit. They use the ripe fruit. That means that fermented the fruit, and then they make the ripe fruit. Use the ripe fruit and make a juice. And after they make the fermented juice, they use that juice to make a formulation, because sometimes they can make a formulation and apply, put some coconut oil and formulation, and then they use it for topical, for reduce pain, and for other kinds of things. So earlier studies show that when in terms of phytochemical, they found there are coumarin, there are iridoid, endoquinone. If you read the literature, there's a lot of compound isolate from noni, even the leaf, even the plant, even the fruit. So this next show you some of the claimed biological activity later on. So this will bring us to why we want to study noni fruit today, and I present it to you here for this particular conference. And what do we mean by fermented? What is the meaning of this? Do we do something during that process? So particularly, we're gonna focus on two important targets because we want to study the cancer activity, cancer chemoprevention activity. We're going to study nuclear factor kappa B and also quinol reductase, and some objectives result discussion before we move on to the next project. So this slide giving you some big picture about when we're talking about carcinogenesis, we know it's a multi-stages. So when we got cancer, it means it's not happening in one day. It involves the different stages: initiation, promotion, and progressions. In the initiation process, what's happening is when the normal cells exposed to pro-carcinogen. So this pro-carcinogen could be from the gas that we expose to, from the chemical we expose, and even bacteria and virus. Activated the cytochrome P450, it will induce this DNA damage, changes normal cell to mutate cell. So this is initiation process. The next, during the promotion process, something happening with this mutation cell. It involves gene mutation, proto-oncogene, tumor suppression gene. Finally, it will become a mix with B9 and also some tumor. Okay. Finally, go through this angiogenesis process, program cell death. If we call progression, it will form malignant and tumor cell. This concept of cancer chemo prevention is not new. It's early already introduced by Michael Spawn back in 1976. So today we are going to study these two important factors that from the noni. Okay, so again, this slide, most of us already know the cancer. If we see some cancer cell with this characteristic, when it has invading apoptosis, the cell not go through the death. Self-sufficient sufficiency in growth signal, the cancer cell continue grow. Insensitivity to anti-growth signal, this cancer cell cannot be eliminated, continue grow. And sustained angiogenesis, limitless replicative potentials, and tissue invasive and metastasis, all these are the characteristics of cancer cell. However, nowadays, they find out that one more important hallmark of cancer is inflammation. Inflammations mean cancer-related inflammations is because of important factor that contribute to cancer. So with that, we'll bring you to cancer-related inflammations. How do we study this mechanism? Therefore, we need to look at nuclear factor. Kappa B is a transcription factor. It's a protein that common in our body that regulate a lot of signaling pathway in different type of cancer. Usually, they are a good protein, but however, when the signaling pathway go wrong, abnormal, so that these pathway keep going on, produce abnormal and a lot of NF-kappa B protein being accumulated. Okay, so involved in tumor initiation and progression in the tissue. So it is important for us to study the compound that can inhibit this nuclear factor kappa B. Another one is quinol reductase. When we lost this enzyme, that means that the cell is no longer get rid of the cancer, more vulnerable to cancer, and also cannot do detoxification. So these are the two important biomarkers we are going to study in Noni. So now we are going to take a look. Why do we still need to study this fermented Noni juice? Noni juice is found in Thailand, found in other countries. What is it new about it? As you can see, sometimes some Noni juice fermented in Fiji have different darker color, brown color, red color. This very nice package, you don't know what is the color inside. All kinds of Noni juice that clean, health benefit, marketed, but without standardization. And if we are going to ferment our Noni juice, sometime when we pick up the fruit, we will mix the unripe and the ripe one. As you can see here, there's no scientific data for physiological of the Noni fruit ripening. We do not know what stage of the fruit they pick to do fermentation. And also we do not know what is inside the chemical responsible for this color. And what is this, during this fermentation process, is there some bacterial changes? There's no standardization. These are the questions we ask. And scary part is some company, when they fermented the juice, they say healthy juice, they claim, but they don't even pasteurize it. During the fermentation process, some bad bacteria that can cause harm to us can create it during this the fermentation process. So as you can see in this diagram here, if you do not control and regulate this fermentation process, it can generate different things, and we do not know. So processing inconsistent, no standard. Traditionally, some use ripe juice, some use raw juice, some raw food, so it's kind of like confusing. Okay, so our hypothesis is we think this juice production through the fermented method has good activity. And then later on, we want to find out what is a compound in that we want to study the cancer, anti-cancer and cancer chemopreventive activity. So that because of this hypothesis, we come with these three aims to prepare the juice, organic and also the fresh juice. We isolate the compound. Later on, we want to study the active compound and study these two different biomarker. We mentioned it earlier. Of course, the project cannot be by myself and with the collaborator. So as I mentioned it to you earlier, Big Island is have this volcano activity. So this island is full of very acidic soil that is the Noni fruit is easy to grow in this kind of environment. Plus we have a lot of rain. So as you can see here, when we pick this fruit, it is ready. They are ripened already, but not too soft and they're still hard. So we sterilize the jar and then we fermented this, give some space. The fruit will fermented for 16 days in the sterile jar. As you can see, if you do the fermentation correctly, the solution come out is clear. It's not like earlier, some of these we cannot see what is going on here. Okay, it's clear at the beginning. So what is happening that during this fermentation process, as you can see, this is the measurement of the carbon dioxide increase in the 14 day. After 14 days decrease. However, the oxygen level decrease. Interestingly, for the pH starting with 10, the pH also reduced on the 14 day. That means it's more acidic. decline in the juice pH and soluble solid, as you can measure this. So in terms of the bacteria, we want to study what is going on in the bacteria. So consistently, when they're doing the different batches of the Noni fermentation, this is actually nobody discovered this before. It discovered in this study that is this fungus called Mucocystinoi is found in this Noni. This fungus during the fermentation process, it is actually the highest of these fungus in the 14th day, and after that it reduced. Okay, peak population at 14 day. After the 42 day, what is happening? Well, after 42 days, you will have this not a good bacteria grow up. So basically, we are saying that these fungus doing the natural fermentation actually protecting the juice from being bad. Okay, so because we want to have more juice to storage, so we continue the study until 21 days, and then the juice yield will reduce after 21 days. So this is an important message. If we want to ferment the juice for 14 days, that's best. But if you want to get more juice, up to 21 days, so then we can harvest the juice and then pasteurize it and then do it for study. In terms of the organic acid, we are wondering what kind of a component is in the juice. As we find out some of the acetic acid, tartaric acid, ascorbic acid, vitamin C. So the tartaric acid is about 1.4 to 2.8 milligram per ml, almost like the grapes. And it is this trace amount of citric acid, and butyric acid that give this noni that kind of interesting taste. Okay. So when we finish our IN1, as I said earlier, now we are going to do, we have this fresh juice, we want to also extract in the organic, and then we want to send for testing. From that one, we are going to study the compound. So what is happening here, we pasteurize this juice, and then the fresh fermented juice, pasteurize it, and then we're ready to send for the collaborator to do testing. The juice also, another bucket, 10 liter, we extracted with organic solvent like ethyl acetate and also butanol. And then we have these two different organic solvent, and then we also send it to the collaborator. Unfortunately, in Hawaii, our lab do not have animal facility. We have to work with a collaborator in the United States in the mainland. So what is happening? This is a collaborator. He is in Clemson. He has this animal facility. He is studying this fresh fermented noni juice, as you can see here labeled F and E. He also studied with the control phosphate buffer saline, and he also used the ethyl acetate and also the butanol organic layer, and he challenged the mice. The mice injected with 0.2 ml of noni juice, fractions of buffer, 2 weeks and 1 week before you give the mice this kind of tumor. So this is what his study showed that with this control, the mice not survive, as you can see here. And also interestingly, with the ethyl acetate is also not good, except the butanol fraction. But butanol fraction of the noni juice and also the fresh noni juice gives 75% and 100% survival for the mice. So this in vivo study clearly show that the active component to have this cancer chemopreventive and anti-cancer property is in this butanol and also this noni juice and this fermented noni juice. So next we want to know what is the active component in there. So it's actually very challenging to do bioassay guided fractionation using the animal model, because if you have small fraction, how are you going to do with the animal? We are trying our best to study what is the active component, and we subject our butanol, because here it shows the active component is in the butanol, right? So we put the butanol organic layer into the column, and then as a natural product chemist, we run the column, and then from that study, we found a few compounds. I just show you 7 compounds here. As you can see here, the compound number 7 is it's kind of flavonoid with the sugar, because butanol supposed to contain a lot of sugar. Compound number 1 is actually a fatty acid ester. Compound number 2 is a new derivative, also ascorbic acid. Compound number 3 is an iridoid, and 4, 5, 6 is also iridoid. So these butanol fraction contain the iridoid phytochemicals in here. So this next few slides, I may go a little bit faster because not enough time. what it's going to show you is how do we determine the structure and able to get a final paper is when we have the pure compound, we determine how many carbon, hydrogen, oxygen we are using high resolution mass spec. In fact, we have this equipment in our college, so this is how we determine the structure. Next, we are running with the nuclear material resonance, running the proton MMR with the protons and carbon and MMR, we are able to determine the structure. So this slide show you the proton MMR chart, and briefly I just show you the around 0 to 10 ppm here. Around this region show you the presence of the sugar, and this skeleton definitely is an iridoid. The next slide show you is the carbon MMR. So which carbon signal is responsible for which CH3, as you can see here. This is the CH3, this is a CH2, etc., etc., like that. And the carboxyl group is in the high field, okay, upfield. So with that study, we are able to publish a paper in a journal of natural product. We did publish this fermented noni juice. So this fermented noni juice is the edible and the drinkable juice can be sold in the market. Next, we want to study, now we have this individual compound. We want to study, compare with other components, and even with the standard, even with the control. What is the compound responsible for NF-kappa B activity and also quinol reductase? So from our earlier study, some of the compound not show activity, but some of the compound like compound 2 and 7 like the flavonoid compound 2 is the ascorbic acid derivative, show high inhibitions and also not toxic. This means not toxic to the normal cell during inhibition of nuclear factor kappa B. Remember earlier I say NF-kappa B is responsible for create inflammation, contribute to the cancer. So compound 2 and 7 are those iridoid compound, and also they are good in showing that, and also compound number 6, compound 2 and 6, those are the compound 2 and also 4 and 6, yeah, those are iridoid that show good quinol reductase induced activity. Yeah. So because of that, we need to do this testing for these 2 biomarkers as I mentioned it earlier. And this slide basically explains to us when we expose to pro-carcinogen. So for example, like this kind of component could be from cigarette, could be from the environment. It will change it to, because of cytochrome P450 changes component to benzopyrene 3,6 quinone compound. This compound create free radical, it will create DNA damage. This is the beginning of cause the DNA damage initiation process for the cancer. However, if we are able to enhance quinol reductase activity, it will bypass this process. It will change this component, not going to create benzopyrene 3,6 quinone. It will only create conjugate and excretion of this compound. Therefore, if we can detoxify of carcinogen using phase 2 enzyme like quinol reductase involved in detoxification, this is how the phenomenon of the noni juice component can enhance quinol reductase. Therefore, it can detoxify the cancer component and also chemical toxicity. Okay, so this iridoid is the component that we found out is responsible for the activity. Not only that, the iridoid show that also activated a lot of antioxidant enzyme in our body, like glutathione, this kind of enzyme in our body, catalase, superoxide dismutase, and also inhibited this MDA that occur naturally as a biomarker for oxidative stress in the liver cell. So far our study show that we established a method about harvest the ripening fruit, and then we also first time we found out this fungus that is doing the fermentation process, and we also show that 14 days is enough. However, if you want to get high use of juice 21 days, we also recommend to pasteurize the juice. Pasteurized juice still show anti-cancer activity and cancer chemoprevention activity. We show you the data already. And the 3 of the compound now, I show you the earlier structures. Some of the compounds show NF-kappa B activity inhibition, and also some compounds will induce quinol reductase. So for our future study, we want to standardize the juice. We are going to use HPLC, and eventually we want to have a possible way that standardize this product for other potential use. So I hope I'm not too fast because I just want to make sure I have enough time to finish up the next presentation. So the reason why I talk about this project is still new because I guess most of you have Noni in Thailand. So as a keynote speaker, it may be nothing particular new about Hawaii. So this project is a collaboration project with Dean Thanapat, also with College of Pharmacy with Dr. Supaki, and also fortunately in our University Hawaii Hilo, we have marine science major, and we also have master student and undergrad student. The professor Akala Metalik, they love to collaborate with us. As you can see here, these show you the one of the algae, and this one is a red algae. This one is the green algae. This is also the green algae. They're just a different shape. And this one is a brown algae because the environment full of different kind of algae. So it's interesting project and has a lot of great potential. So particularly, if you ask me why we look at antimicrobial activity, because as I say, our lab not possible to do complicated things like we cannot even do COVID-19 in our laboratory right now. So we are looking at the infections, MRSA. There's increasing methicillin-resistant Staphylococcus aureus infection and drug resistant. We all know Staphylococcus aureus, most common bacteria involved in human infection in the worldwide. So MRSA listed in the CDC in the threat list. Okay, so Hawaii has twice the national average of MRSA infection. With native Hawaiians are more prone to Staphylococcus aureus infections. Therefore, it is not very attractive. We want to find this antimicrobial agent from these rich natural resources. Okay, so what do we mean by community acquired? So it's not only the MRSA list in the hospital clinical isolate, because when people go into the beach, if they're exposed to the community acquired strain, when they go exercise, sweating, they can easily acquire the community acquired strain as well. In the military, in the daycare, especially right now, COVID is getting more complicated. So with this condition, community acquired MRSA, not just hospital acquired, it becomes a serious issue. So these are the things, therefore, I need to work with Rancid because they also have facility related with the different strain in the hospital. So now we back to in Hawaii again. Limo means the algae, right? The ancient Hawaiians say there are more than 63 edible species. However, there are only very little literature study about Hawaii seaweed bioactivity. We call it macroalgae. So I'm going to focus on the edible first because sometimes it's not easy to collect this algae. 30 grams is okay. When you get larger amount, could be very challenging. So I'm going to focus on these three, show you some of the primary data. I showed you earlier this one, they use it for put under the raw fish. It's called the red algae, and this one is Algae. We also buy it from the KDA. They also grow it in the farm and they sell it. And the other one, this one is a brown algae. Okay, so I want to share with you. I know today the talk about we are here is because of a pandemic, right? COVID-19. I want to show you some very interesting literature why we need to study this algae. So Hawaiian marine algae had widely varying compound with antibacterial activity. Not only that, so our earlier study showed that these red algae and also the Algae and other species will be collected. And we are going to do two methods. One is organic, one is called a water extraction. So I give you this connotation here. Water extractions mean we extract the polysaccharide compound. It's not just natural product, it's polymer. So why we need to interesting study that? Because right now there's a recent literature show that these polysaccharide extracted from this Saccharina japonica show antiviral activity in this viral cell infected with this COVID-19. And not only that, the highly branched structure even show EC5083 nanomolar and more potent than the reticinin. You can go and see the literature in there. So this is definitely very interesting, and even some earlier studies show that this fucoidan polymer might be interfere with the spiky protein on the surface. Okay, so now back to our lab. This one is the literature. Back to our lab, we collect this and identify this algae. We have a marine professor there locally. We have students collect this anytime when the algae is available with low tide. Bring it back to our lab. We use organic method to extract it. We also use water extraction. Right now, I only show you acidic conditions and sulfide, this high temperature, because we worked out this previously. I easily can change it to different condition and extract it and create another extract. Then next we will going to test it. So far we are only test antimicrobial and antioxidant activity. I didn't show it here, but in the future we can also test antiviral activity. Only using viral cell in my lab, but when we have to do COVID-19, there only one lab in Honolulu has the permit that I have to send it to there. Unless in Rancid University, somebody willing to work with me, I can send the compound or the extract to Rancid to do the COVID-19 testing. So I'm not going to talk about the strain in detail. We use MSS, Methicillin-resistant strain. We also use MRSA, Methicillin-resistant strain. We also have strain isolated from Hawaii, and we're going to use Kirby Bauer and also the MIC method. And based on that, we design the experiment and find out which MIC are active. I have only one slide to show here. It's very preliminary, but the data is so robust, I forgot to attach the literature. Is if you have the lye, by using this dye, you have the sample incubate with the bacteria. The next day, you can add the dye. If you have the lye cells, if you have the lye cells, it will change the purple blue to the pink color. That means you have the lye bacteria here. But if these bacteria are dead with this concentration on the ampicillin, and it will stay as the purple color. So this row show the Limu Kohu as very preliminary result. We run in triplicate, and also this one is the edible ogo. Again, this is still the extract and it's not the purified compound. So because running out of time, I'm sure some of you remember who is Ashley. She been to Rancid. She also studied this. This is the kelp people can buy in the local store and make it into the noodle and eat it. We also found some very interesting activity. Fucoidan, we extracted, and then we purified the fucoidan. These are some earlier work. However, it is this branch structural fucoidan. It's not just the standard fucoidan. Might show the different activity that what I was talking about. We finished this study earlier, and we show some anti-cancer activity for this fucoidan. So here are the references. I almost running out of time. I just want to wrap up a slide with this. Study is with this pandemic right now, we do not know what we're gonna facing in the future. I am sure material having an issue, but you can see algae here, you can see mama kitty here, you see Noni here. The search for naturally occurring cancer, chemotherapy, and antimicrobial agent from nature has been increasing. If we can improving and promoting local plants and marine algae and product potential in Hawaii, it could be helping us as a sustainable sources to possibly have good health and even for food and other things, because we cannot just depend on medication. So acknowledgement to the collaborator and also some of the funding with the embryo, not able to have time to go over that in detail. And if you come to Hawaii, this is a Pacific Ocean. I will show you around, but probably not right now. And you can go over the ocean and pick the limu, and you can see how many limu you can select. Okay, so I'm going to finish up this slide. This is the edible ogo that we tested in the lab. Show the activity, the water extract. Okay, so I'm going to end my slide here. Thank you for your attention.
Thank you very much, Associate Professor Dr. Leng Chang. Now we come to question and answer session. Please feel free to unmute and ask.
Yes. I'm Sasurin from the Faculty of Food Technology, Rancid University. I have some more questions. Why don't you cut noni fruit in small pieces to extract more bioactive compound inside fruit?
Okay, first question. You asked me why I don't cut the noni fruit, right? And extract the sample. Yeah. So what happened is we are following the native Hawaiian healer way. you can cut the fruit, but we want the mucor to ferment naturally. So the purpose of doing this is when we pick the fruit directly, we try not to mess up the mucor. We let the mucor fermented the juice naturally. So therefore, we got this juice, and then we pasteurize it, and after that, we extract it with the organic sample. Because this is the juice where the healer, and even we already showed that it had activity. Yeah, I hope it answers your question. Because at the end of the fermentation, the fruit solid material go down. It doesn't make sense to cut the fruit anymore. After 17 days, we actually harvest the juice. What we do is we squeeze the juice, we squeeze the fruit, get as much as juice we can, but we do not use the solid. The active ingredient is in the juice, because this is how the native Hawaiian healer use that juice to make the topical application as a remedy. I mean, they use the whole plant, but for particularly they use the ripened fruit. I hope I answer your question.
Okay, thank you. Do you add any sugar or specific microorganism for your fermentation?
In this particular case, we did not, because we know that other group also used add the sugar. So we just let the mucor to do the fermentation process. Yeah. It would be nice to comparison. Another student can add sugar, and see whether it speed up, whether it will have similar activity. But we didn't do it as a comparison. Yeah.
And I have some more question. How you control the fermentation condition? Because
control.
How you control your fermentation condition?
Okay. Do you mean temperature?
Yep, everything.
Okay. So what happened is after the jar being sterilized, right? It is actually put inside a room. In this room, actually, you can put them in the room temperature. Okay. So that the lid is actually, because you sterilize the jar, it's not too tight, and you cover the lid. So they are all in a similar condition, room temperature. Yeah. But naturally, some people want to let it expose to the sun. If you see some slide earlier, some people naturally they can under the sun, you will see different color. So for this particular project, they got the juice more or less the same color because they put it in a similar condition. Yeah. I hope I show you. I hope I explained that the pH, the CO2, the temperature, and consistently different jar they found that the mucor. Yeah.
Yeah. Because as you mentioned earlier, that it's quite difficult to separate the raw and ripe Noni fruit. So can you control the ripening or the condition of your Noni fruit before fermentation?
Okay. So very good question. So what is happening is when we pick this Noni fruit, they are at this stage. We don't pick the green one. Okay, you see that? We don't pick the one drop to the ground and soft. We pick the one hard. We pick the hard one. We wash them and let them dry. So not too soft. So they are more or less around the similar timing. Yeah. Interestingly, some people actually explain to us that the moment when the Noni turn to yellow and white, there's also important thing that we need to study. But according to Ayurvedic medicine, but we didn't repeat the experiment. Yeah. So I hope by doing so, we have one way to control it. Yeah.
Thank you.
Other question? So anybody here in Ransit are doing COVID-19 screening? Maybe I need to talk to your team. I know exposed to COVID-19 is not fun. Yeah.
Yeah, I'm the one who enjoy drinking fermented Noni fruit. But I normally add some sugar, some microorganisms, special microorganisms, and cut into small pieces for better fermentation.
How long did you fermented it?
Yeah, 2 weeks.
Oh, okay, 2 weeks. Do you pasteurize the juice?
Yeah. Now for fermented, no need to
Pasteurize.
Yeah.
Okay, I see. But during the fermentation process, do you sterilize the jar?
Yeah, absolutely.
I see. Thank you.
Hello, excuse me.
Yes.
I do have a small question, especially on this topic. My name is Pemadoji, and currently I am doing a master's in curriculum education under Rangsit University.
Yes.
So I do not have a science background, but I'm eager to know this. You are talking about the fermentation, that especially this fruit outside in the sun. I was just wondering, you are talking about the room temperature, and then you are exposing the fruits outside first. And how is the strength of the medicine? For example, if you expose the fruit in the sun and then without exposing to the sun, so what is the medicine values that you can have out of that? Is my question clear?
Yeah. Your question is clear.
Okay.
So let me.
Yeah.
I think the sun will increase the heat.
Oh,
right?
yes, exactly.
Actually, what happened? I want to explain to you. When I hear from the lady Hawaiian healer, they teach me the way that they teach me. This lady Hawaiian healer. Earlier you say that our teacher. They teach me sterilize the jar, and then put the noni fruit jar near the window, so that you have some near the window. Not mean direct sun, not directly like sunlight is about the temperature. Not like this. Okay. However, this process that we are doing, not under the sun. It's inside the room temperature. Inside. Yeah. But this is for experiment to be consistent. So therefore, even this juice, we are harvesting has a lighter color. Because different temperature maybe affect the microbial and the pH, and also this that will affecting the color of the product, I guess. Yeah, as you can see here. Yeah.
Okay.
I hope
Thank
I
you so much.
Yeah.
Thank you so much for your good answer.
Yeah, I hope people will come to Hawaii. We have no
Thank you.
we have a lot of resources.
Thank you very much.
Thank you. As we run out of time, could we move to the next keynote speaker? Thank you very much, Associate Professor Dr. Leng Chang, for your impressive topic.
Thank you.